The overall goal of this protocol is to demonstrate an optimized passive optical clearing method that is suitable for use with human pancreas tissue.
This method can help answer key questions in the neurobiology field relating to diabetes by providing details on the innervation of islets.
The main advantage of this technique is that the clearing method ensures transparency of human pancreatic tissue for use in high-resolution confocal imaging.
To begin, fix a pancreas sample by placing it in freshly prepared 4% paraformaldehyde and incubate the sample at 4 degrees Celsius for 48 hours. Next, chill a flask by placing it in a bucket with ice, and then place the bucket on top of a magnetic stir plate. Make sure the flask is sitting flat and add a magnetic stir bar. Pour 147.8 milliliters of ice-cold distilled deionized water into the flask. Then, add 20 milliliters of 0.1 molar PBS, 20 milliliters of an ice-cold 40% acrylamide solution, 12.2 milliliters of a 16% paraformaldehyde solution, and 250 milligrams of VA044 initiator. Mix the entire hydrogel solution for at least 10 minutes. Next, place a 15 milliliter conical tube in the ice next to the flask containing the hydrogel solution. Pipette